All right, next lecture. This is on biology, section 1.2, the methods of biology. So this lecture is about, first thing to talk about is humans, and most creatures have curiosity. You've probably heard the phrase curiosity killed the cat. This is the quality related to inquisitive thinking, or trying to figure out what's going on, such as exploration, investigation, and learning. So that's curiosity. It is one of the key elements of what it means to be an animal. Most animals have this, cats especially. And most of the time there are a couple of steps that we use to satisfy our curiosity. One name for these set of steps is the scientific method. The scientific method, oh, that didn't work. Hold on, see if I can erase that. Yes. No? Yes. Erase. Erase, I say? Sorry, still getting used to this, how to use this program. All right, the scientific method is the general name we have for the way that we can satisfy our curiosity. Now, there are several steps to this process. Today we'll be splitting this process into 6 steps. We are going to go through each one in detail over the next couple of pages. So we are currently, these notes should be written on pages, let me figure out which page. I can't remember which pages they were supposed to be on, but the 2 pages that I indicated in class are the next ones you need to put this. The next 2 pages are the notes where you should put these notes. So once again, these notes go on the 2 pages that I indicated in class. If I did not indicate it in class, do it the next 2 pages after our discussion in class today. Or I guess after your notes for 1.1. Alright, so step 1. Step 1 is to identify a problem. This is usually done through observing our world. Observing the things that are working, the things aren't working. An example of this is if you went to Guam, an island in the South Pacific, you'd find that the bird population was declining throughout the 20th century, through the 1900s. And this bird population was declining rather quickly in Guam. So that'd be the problem. So our problem here, example problem, is the bird population is declining. Okay, that's step 1, identify the problem. Step 2 is to create a hypothesis. This can be described as creating an explanation that you can test. So if you create a hypothesis, you're creating an explanation that you can test. One of the observations that they made was that there were brown tree snakes in the trees, but very few birds. And so the hypothesis that they came up with was that the brown tree snakes on Guam are eating the birds. This is their hypothesis. It could be tested. They could go make observations and figure out whether this is actually occurring. All right, so that was step 2. By the way, this is not a random guess. This is not something they're just making up. This is based on observations. So they make this decision, come up with a hypothesis because they think it is a reasonable explanation for what's going on. All right, step 3. This is actually has 2 parts. The first part is going to be step 3A. This is where we create an experiment. The experiment, the definition of experiment is to collect information under controlled conditions to test a hypothesis. So that's what an experiment is, to collect information under controlled conditions to test a hypothesis. Now, we need to talk about these controlled conditions, kind of talk about what that is like. If we have a controlled experiment, a controlled experiment is where we have 2 groups. We have the control group, and we compare the control group to the experimental group. Now, one way you might think of this is if we talk about cookies. Our topic is here, it's going to be cookies. A control group might be a standard chocolate chip cookie. So chocolate chip cookie. All right, that's our control group. It's what we're comparing to. Our experimental group might be where we take our chocolate chip cookie and we change one of the ingredients. Like we replace chocolate chips with raisins. Now, some of you may think this is a terrible idea. I don't disagree with you. However, this is one way we could test. Our control group would be the things we're comparing to, our standard chocolate chip cookies. We're comparing that to our experimental group where we've changed something. And we're hoping that we can see what the effect of this change is. Now, you'll notice I did not change the flour and the raisins and the butter. I didn't change a bunch of things. One of the key elements of a controlled experiment is we only change one thing at a time. So I changed chocolate chips to raisins. I do not want to change the flour or anything else. I'm just going to change one thing at a time. All right. Now, we have some names for this. There are 2 different kinds of variables in this situation. A variable, by the way, is something we change. Something we can change. There are 2 types of variables. We have independent variables and dependent variables. Independent variables are variables that that are the only factor that affects the outcome. Dependent factors, on the other hand, are the variable that is affected by the by changing the independent variable. So the independent variable then is, in our case, is raisins versus chocolate chips. That is the thing we're changing. We're changing the thing that we're putting in our cookies. Are they raisin cookies or are they chocolate chip cookies? That's what we're doing. Now, that is our independent variable. Our dependent variable could be many different things. The one that I'm thinking of is moisture. Are these cookies more moist or less moist? Are they softer? You could say softness. This is another potential dependent variable. We could talk about flavor, see if they're nice, whether they're sweet, or if they're bitter. Those are all different things that could be affected by this change in from chocolate chips to raisins. Okay. Once again, the raisins and the chocolate chips are the variable that is the factor that is changing the outcome. The dependent variable, once again, is the thing that's affected by the changing of the independent. So moisture, softness, flavor, sweetness, all could be dependent variables, while raisins and chocolate chip switch between the two is our independent variable. All right, so let's see. That's independent and dependent variables. These are very important for our study of science. We can have a different kind of experiment, a non-controlled experiment. This is usually where we do observation of the world without any control of the variables. An example might be if I was looking at bird behavior. I wouldn't be able to control what predators were present. We couldn't control for the weather. We couldn't control for many different things. So if I do a study on bird behavior, this is a non-controlled experiment because there are factors I cannot control. It's non-controlled. Okay. So the other part of this is that these experiments will then give us data, will produce data or produce information. Produce information of some kind, either numbers. This could be this could be time. This could be temperature. This could be length. Time, temp, length. We could also do things such as mass or area or volume. We could also get information on behavior, stuff like that. Those are all kinds of pieces of information we get. All of these, in science terms, the term we use for this is data, or experimental results. So if you hear me use the word data or use the word experimental results, those are both terms that I use to describe this information we get from experiments. So once we have this data, we go on to step 3B. Step 3B of the process. Here we analyze the data. Z. Okay? When we analyze the data, we figure out what it means. We ask questions such as, oh dear. We ask questions such as, was the hypothesis supported by the data? We ask questions such as, are more data needed? Or how do the results compare to other studies? Those are all things that we might ask when we're trying to analyze the data and decide whether it's meaningful or not. All right, next we'll talk about step 4. All right, step 4. Moving on to the next page. Don't forget that once you use up the 2 pages of your notebook, you need to put a piece of paper at the bottom, either stapled or nicely taped in order to expand the amount of space you need in case you need it. So step 4. Step 4, reporting the results. It is very important that you share your results with people. There are several ways that we do this. We share results by publishing them in scientific journals. Or in our case, in this class, the way we will do it most often is by talking about them with fellow researchers, or in our case, fellow students. So that's step 4. Step 5, one of the reasons we report the results is that we need to verify the results. We need to get a clarification as to whether they're actually right or not. If you've heard the phrase, many hands make light work, it's the same idea here. One of the things that's important is that we use what we can say, many brains make light work. And so by sharing our information, we're allowing other people to check it for us and to help us figure out whether we are correct. So having other people help us verify if we are correct, verify our answers. All right, let's see. Next, we will talk about step 6. All right, step 6. Oop, not STED 6. Step 6. Right. All right, step 6. What we do is we create a theory. By the way, a theory is an explanation of a natural phenomenon that is supported by a large body of scientific evidence. And this evidence has been obtained from many different experiments. Oh, sorry, many different experiments and observations. Theories specifically are an explanation. We explain how things work, why things work, that kind of thing. Theories are not something we come up with easily or kind of flippantly, where you're not really trying very hard. They're something we spend a lot of time developing because they are supported by a large collection, a large body of scientific evidence obtained from many different experiments and observations. These theories then help explain scientific laws. Now, a scientific law, by the way, let's define that real quick. Scientific law is a something that is true about the universe. You might think of scientific laws as things like gravity or the speed of light or the law of conservation of mass. All of these things are scientific realities, truths about the universe, things that we are very certain of. These are like truths or facts. And the why, but the why, the how, the when, not the when, but the why and the how is related to theories. So once again, just to clarify, theories are why and how. That's the kind of things that theory answers. And then a scientific law is a fact or a truth. That's the two. That's how those two are different. So this is how you would go about the scientific method. Once again, step 1: identify a problem by observing, figure out what's wrong, figure out what you want to figure out. Hypothesis: create an explanation that you can test. Step 3: do an experiment and collect information under controlled conditions with a control group and an experimental group. One is changing, one is not. We have the independent and the dependent variables. These are the variables that we can modify. Independent is variables that are the only thing that affect the outcome. Dependent is the thing that is affected by changing the independent variable. And then we analyze the data we've collected, the information that we've collected. Then, once we've done this, we report the results to others, either through talking about it or by writing it down in some way. We verify the results by sharing the material and having other people check it for us. And then, after many rounds of experiments and many attempts at figuring out how to handle the situation, the problem we are facing, we create a theory, which is the how and why that explains the truth or fact that we see as the law, scientific law. All right, that is your lecture for 1.2. Good luck. Have a good evening, and I'll talk to you later.